Hello, everyone, and welcome to today's webinar, Comprehensive Characterization of Etanercept using the ZenoTOF 7600 system. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SCIEX. To learn more, visit sciex.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Xuezhi Bi, Associate Professor, Head, Analytical Science and Technology, Protein Analytics, at the Bioprocessing Technology Institute, A*STAR Research Entities. Dr. Bi, you may now begin your presentation.
Thanks for the kind introductions. I would be happy to present our experience of using the new ZenoTOF 7600 for the comprehensive characterization of a fusion protein, Etanercept. So this is the outline. I will briefly introduce our Institute of BTI and the innovative ZenoTOF MS. Then I will introduce the background of the Etanercept and use the new mass spec to characterize its critical PTMs and glycan profiling. So our BTI is full name is the Bioprocessing Technology Institute. It's a national academic research institute funded by the Agency for Science, Technology and Research, in short, A*STAR. We provide seamless integration of end-to-end bioprocessing capabilities for academic and industrial partners. Like biotech companies, our core competence developed in BTI since its inception is integrated capability in biologics development in all aspects of value chain, such as product innovations and rapid cell line development and protein free chemical defined media development, process development and scale up, and downstream purification process to innovative analytical science and technologies for rapid streamlined product and process characterizations. With this kind of competency, we develop innovative technologies to facilitate biologics development and manufacturing, enabling product and production of next generation of products such as antibody, drug conjugate, bispecific antibodies, CAR T cells, and extracellular vesicles, such as including exosomes, virus-like particles, viral vectors, and also the vaccines. So the more and more next generation biologics with like multispecific antibodies and mixed antibody cocktails, and the vaccines, the fusion proteins are emerging. So the complexity of the next generation biologics increasingly in challenge for comprehensive characterizations. The following common critical quality attributes that need to be comprehensively characterized is such as the molecular integrity of the sequence verifications, the amino acid oxidations, deamidations, isomers, and also the unlinked or linked glycosylation. So disulfide linkages. So these are common CQAs we need to address. Here are the key features of the ZenoTOF 7600 system. They can overcome the duty cycle deficiency, and also they have sensitivity gain up to 5 to 20 times. It have the tunable fragmentation pattern for all molecular types, the low-for electron activated dissociation, EAD. So all the MS mode can be enhanced to be 5 to 10 times with the xenotrapping technologies. So with the MS scans, rates can up to 103 Hertz. They improved the DDA with a high resolution MRM, wide intra and inter-scan dynamic linear range. With this, no other system at the routing alternative fragmentation like this. So detection up to 20 more ions in every experiment, and also the spectrum fragmentation to unlock new perspective for every molecules in the every experiment. This is really a good advantage of the ZenoTOF. As shown in the upper figure, the MRM high-res intensity and the signal-to-noise ratio gain of the peptides is increased by 5 times between the ZenoTRAP on and off the blue and the pink colors. So in the lower figures, so we can see the ZenoTRAP gains is more the mass over D dependent. But in the figure on the right, when the ZenoTRAP turned on, the continuous ion beams pass through the ion gate according to the mass of D. The ion pulsing in the TOF accelerator facilitate the TOF detector achieve the enhanced peptide MS duty cycle from 30% up to 90%, and the sensitivity gains by 5 to 10 times enhancement. Etanercept is commercially sold as the embryo. It's produced in the Chinese hamster ovary cells. They used to treat autoimmune disease. As shown in the right, the molecular structures. Etanercept is the Fc fusion proteins in combination of TNF-alpha receptor 2 with the Fc IgG antibody Fc concentrated regions. It contains the highly glycosylated complicated PTMs. In addition to the traditional antibody, the glycans in the Fc regions, there are 2 unlinked glycan sites here, and also up to 13 all-linked glycosylation sites in the TNFR region. Therefore, the comprehensive characterizations of these complex Fc fusion proteins could be challenged on the traditional mass spectrometer. So based on the limitations of the current status of the mass acquisitions, firstly, we took the middle down strategy to measure the intact mass of subunit level. Here is the intact mass analysis of the convoluted spectrum after decylated and the reductions. So as treated with sialidase, so resulting mass peaks are comprised only glycans, glycan variants, so lacking of the sialic acid. So they be bear in mind. So therefore, we observed all the most abundant glycoforms in the spectrum. The top 2 spectrums, about 50 lying kilodaltons here, is most likely carriers of the compilation of 147 G2F and 171 G2 and the Fc regions G0F. This is the most abundant one. In the compilation with 9 core O glycans or 10 core 1 O glycans in the 2 major peaks. And the next most abundant one is the compilation of the 3 glycopeptides with the Fc regions G1F. The rest are similar, but the 9 core glycans and the 10 core glycans compilations. These are the 2 peaks. The other compilation, the peaks are from the different compilation of the 3 glycans and or more than 7 core 1 O glycans compilation. The different peaks are showing here. So the major peaks shown here are the proteins. The fractional abundance of the 3 N-glycan sites and O glycans will be discussed shortly with the peptide level, glycopeptide level. Now we are working on using the bottom-up strategy to characterize with the peptide mapping. So for the peptide mapping, so we found the standard, the guanidine DTT reductions, so the denaturation, and then we follow the alkylation with iodoacetamide. And after that, buffer exchange dilutions before enzyme digest. So according to the sequence and enzyme specificity, we choose sequential multiple enzyme digestion strategy to achieve the best sequence coverage and the proper size of a peptide to be analyzed. The sequence confirmation of the leucine isoleucine isomers is important because the residues in the variable regions, especially in the CDR regions, may result in substantial loss of antigen binding affinity and the specificity of the antibody. So the regulatory agency, FDA, requires confirmation of a molecular integrity of the 100% sequence accuracy. So this includes definitive confirmation and differentiation between the leucine isoleucine residues. Normally, the leucine and the isoleucine have different, generally considered indistinguishable by mass spectrometry because their molecular mass are exactly the same. Currently, the DNA sequencing and Edman degradations are used for this experiment, but are inconclusive, but also time-consuming. EAD, on the other hand, can directly generate fragment diagnostic ions for leucine and isoleucine to differentiate the isomer pairs. As shown in this slide, the leucine producing 43 Dalton Z43 ions, and while the isoleucine producing Z-29 ions. So here's the example of using EAD diagnostic double ions to discriminate the leucine and isoleucine. The upper spectrum peptide with 2 leucine residues inside, as the Z43 fragment ions are present in the Z3 and the Z6, the ions position there, for we can clearly identify them both the leucine from the EAD and SMS. From the lower spectrum peptides, we have found that both leucine and isoleucine present in the peptides. But we found only one Z43 and also one Z-29 in the spectrum can provide an ambiguous identification of both leucine and isoleucine in the particular peptide sequence. So another isomeric amino acid, aspartic acid, and isoaspartic acid. This differentiation is also a common challenge in the deamidation isomerizations of the critical quality attribute characterizations practice. So the different isomer amino acid sequences have a potential impact on the target binding. The differentiation is also of particular importance. Generally, both aspartic acid and isopartic acid can be generated from the asparagine through the deamidation via succinylamate. And after long time storage, aspartic acid can also isomerize into aspartic acid. So CAD can provide deamidation site, but you cannot differentiate both isomers exactly. So as shown in the figure, the cartoons, so isoaspartic acid provides different fragmentation pattern under EAD because of the existence of the methionine and in the backbone. So it generates the diagnostic signature ions C plus 57 and Z minus 57 instead of regular C and Z ions for the isoaspartic acid. So this figure shows the XIC mass spec in the left and MS mass spectrum in the right of the same peptide sequence, but with different deamidation status and different retention times on the spectrums. So non-deamidated peptide eluted earlier on the bottom, and followed by deamidated isoaspartic acid on the top. This is the latest one. And the last one, elution time, is the deamidated aspartic acid in the middle. So among the 3 spectrum, only top one contains the Z3 minus 57 and the C1357 diagnostic ions. So we can conclude asparagine N is deamidated to aspartic acid. So therefore, we can use the retention time and EADMS diagnostic ions to differentiate isoaspartic acid and aspartic acid with the really high confidence. So let's come to the unlinked glycan profile. Consistent glycosylation are critical for efficiency and safety for therapeutic glycoproteins. Unglycans were known to be a CQA in biotherapeutic characterizations. As glycans possess many additional structures, such as the variable sequence, linkage, branching, etc. So in the traditional CID, the glycans will be dissociated prior to peptide backbones. So predominant fragments are oxalium ions, which are not ideal for glycan localizations and further identity of the glycans. So there are limited peptide backbone information is obtained by the traditional CID. But with the EAD, we can maintain the integrity of the glycans during fragmentation, which is crucial for obtaining the localization and the glycan identity. So this is the Etanercept with the fusion region, the 1317 locations. So in addition to major abundant G0F, G1F glycan is traditionally the relatively low abundant glycopeptides are demonstrated here. Here the upper one is the spectrum and glycopeptide with the -5. Normally you're challenged to detect by the traditional one unless we use the release glycans. So this 4.6% functional abundance. The location of 317 glycan site can be determined by C series ions, C line, C11 and C12. All the C ions can be detected and for -5 glycan modifications. The lower spectrum showed if at lower fractional abundance up to 0.014%, we are still able to confidently identify the N-glycan site from the C series glycans of C line, 10, 11, and 12. The positions of the instrument provide excellent spectrum quality, even for this trace amount of glycopeptide with symmetric branching glycans with 2 sialic acid substitution at the terminals. Here are the summary table of the IgGFC region 317 glycan profiles. There are total 10 heterogeneous glycan structures observed in these glycopeptide. In the protein matrix build softwares, we can automatically heat map, visualize the display of the color-coded abundance with the most intense color reflect the dark colors, and the lower abundance will be the light colors. G0F is the most abundant glycans, species up to 57 fractional abundance across the 5 replicates. Second abundance is about G1F, the 30% fractional abundance. This is really corresponding to the total intact mass fractions. So the result of the top 2 peaks we already observed in the intact mass analysis. So different from the FC region. So the TNFR region of the 140 line have a much more complicated glycan distributions. Most of the N-glycans are sialic acid modified. The G2S1 in upper spectrums and the lower spectrum has a asymmetric sialic acid branching glycans. Even with the sialic spectrum, good quality spectrums produce the localization information according to the series of C and Z ions. We can see closely on the figure. And the upper G2S5 fractionation pattern is about 25%, while the lower one only 0.13%. So we're still able to detect glycans and also accurately localization of the glycan site. This is the 140 line site of unlinked glycosylation, but with heterogeneity of the total 17 different type of glycan structures is observed. So earlier subunit mass analysis doesn't cover this glycan in detail because with the sialic acid modifications, but we captured the complete structure with unlinked glycans here because this one we didn't remove the sialic acid. So fractional abundance of each glycan type are consistent across 5 replicates according to the report here. The different replicates we can detect. And similarly, one site has also 17 heterogeneous glycans. The most two abundant is G2F glycans with 1 or 2 sialic acid residue attached. By using the glycopeptide EADMS spec analysis, we are able to identify and relative quantify individual glycan fractional abundance, even at a very low trace level of glycopeptides. And also, these 5 replicates shows excellent reproducibility. As mentioned earlier, the Etanercept TNF regions contains up to 13 all-linked glycosylation sites. This, in contrast to unlinked glycans, all glycans don't have the single consensus sequence for potential glycosylation site and also have no kind of structures for prediction of the glycan structures. So it is more heterogeneous and often clustered together. Most of the high density and particularly the serine and threonine clusters. And also all linked glycosylation sites are often labial and complex. The characterization has been a challenge for years due to many reasons. So first one is unable to predict modification site, and frequently multiple potential site consecutive sites are in the tandem sequence in the same peptide, which cannot be separated by the spatial enzymes or most of the time, the most abundant one is the non-modified glycosylated peptides. So CAD normally cannot maintain any glycan information in the MSMS spectrum, not being able to ambiguously identify the site of localization in most of the case. On the EAD can maintain the integrity of glycans and fragments, which is crucial to obtain the localization and the glycan identity. Here's the example of the EAD MS spectrum of one of the peptides, 37 amino acid residue long. It's a triplet peptide. There are potential 11 C and threonine all-linked glycosylation sites. Yeah, so many sites. With clearly the spectrums, we can identify the C and Z series ions. For example, the C500 Dalton's modifications to confirm the location of all glycosylation site is threonine 208 and threonine 212. These two occupied sites with sialic acid substitution from the DDA, EAD, MSMS. We also able to observe glycopeptide with other 5 all-linked occupied sites with a single or double glycosylation site as reported before. Similarly, we could also couldn't see simultaneous 6 to 7 serine threonine residues occupied by core 1 all-linked glycosylation site, either without removal of sialic acid and attached to these glycans. So this is a summary of the 10 sites of all-linked glycosylation identified using the DDA and the EAD without enzymatically removal of sialic acid. So these 10 sites of all-linked glycan distribute among the 6 different peptides fragments. So as shown in the table, all glycans are predominantly core 1 hex-like subtype substituted by 1 or 2 sialic acid residues. So for the serine 182 and the threonine 184, earlier reports couldn't because pinpoint exactly all glycosylation site confidently. But in our study this time with the EAD, with the trypsin as pyridine combined digestions, we are able to detect both the serine 182 and the threonine 184 all glycosylation site successfully with the DDA EADMS fragmentation with the different iron series. To summarize, the versatile EAD can confirm the small or terminal peptides differentiate aspartic, and isoaspartic acid with direct MS instead of inferred from the chromatography retention time, confidently quantify and extensive sequence coverage with positional identification of even low abundance glycan species. So the easy and tunable acquisition combined with a streamlined workflow from PMI to BIOS software enables the comprehensive N-linked and O-linked glycan characterizations from the complex FC fusion proteins. Most importantly, I need to mention the innovative ZenoTOF system. So it equips us improved tools to characterize complex late generation biologics. So firstly, the ZenoTOF increased MS sensitivity up to 5 to 10 times and improved duty cycles up to 90%, significantly improved the quality of the EAD and the CID spectrum. Secondly, the DAD is an approach and this highly producible electron-based fragmentation method. This tunability of EAD provides access of multiple modes of electron-based fragmentations, and also the SCIEX software combined with the proteomics and BIOS, so first facilitate an efficient platform to analyze the late generation biologics. Here I would like to acknowledge these people who contributed the work of these presentations. First, I need to thank the Xuezhi Zhang and really working directly from very beginning to the end of the experiment and the data analysis. So, and also I would like to thank my team members, Dr. Ko and Dr. Pang and Ivy. So I also would like to thank Steve Tate, Elliot Jones, as well as the Jason Liu for their multiple years of BTA SCIEX collaborations. Last but not least, I would like to thank you for your attention for my talk. I would like to happy to answer any questions you may have.
Thank you, Dr. Bi, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Dr. Bi indicated he was having some audio problems. Dr. Bi, are you ready for the Q&A? It looks like we have already received quite a few questions from the audience, but unfortunately, we have lost our connection to Dr. Bi. Please submit any questions you have for the presentation, and we will send an email communication with all of Dr. Bi's responses. We sincerely apologize for the technical problems. So with that, I will thank you again, Dr. Bi, for your time today and your important research. We would also like to thank LabRoots and our sponsor SCIEX for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.